conduct the T-cell rate. And after 2002, all the criteria and the set of hospitals are the same. So I will show you the data on that. So for our surveillance, there's a major issue about Candida Tropicalis. And one is Tropicalis is one of the most common non-African Candida species. And then, if I don't want to bother you for business, but if you see the red lines, red color means resistance. So Candida tropicalis have a higher resistance rate compared to other species. So then, for me, logically, we want to know is whether those resistant isolates are genetically related. So we use MST to analyze genetic relatedness, and surprisingly, the resistant isolates were closely related and belong to clade 4 after 2014. That's why I want to ask the evolution expert is, before we didn't detect that, but after 2014, it's continuously and predominant. So, of course, we want to know what's the advantage for clade 4 to be so predominant. And there's one I would like to show you is the resistance. Fukan resistance was marked by red R. And here is genetically related, the clade 1, 2, 4, or 5, or others. So the take-home message for this slide is that the clade 4 doesn't have growth defect. So it's not like the other resistant strain has a growth defect. This is not. And all the clade 4 but not other resistance in other genotypes has a mutation on the target of Ego11. And the funny part, I think this is very interesting to you guys, which I don't think is possible. So, but that's happened. On tip epoch of Candida tropicalis, one copy of chromosome have wild type Ego11. Another copy of chromosome have a mutated Ego11 and also high copy. So this is why they have a wild type copy of Ego11. That's why they have no growth defect. But they have multiple copy of mutant one, so they can very easily adapt to the situation that have the other drug. Let's make this become the super one among the Tropicalis resistant Tropicalis. And it says for the introduction of the previous talk, so I don't have to go through very detailed. This is a possible compound after taking drugs. First, of course, we take a